Each year, thousands of students come to the university without any information on scientific research activities which are linked to education. It is possible to inform more on this topic.
Or whether not. So they're looking for these particular molecules.
And how we do? Comment on pratique? On fait la technique d'électrophorèse. Particulièrement, c'est une technique que nous utilisons beaucoup. Electrophoresis, you know what is electrophoresis?
Electrophoresis is on a bone cockpit and on the feet. So basically in electrophoresis you have molecules, and according to how big the molecules are, they move at different speeds. Dala, Rita? So, and you've got an electric field, that's why it's called electrophoresis. So which ones do you think are going to move faster, the small ones or the big ones?
The small ones.
The small ones. So the small molecules move faster through the field, and the big ones get left behind, and you get these different things. Have you ever heard of DNA fingerprinting?
Yes.
Okay, DNA finger. If you take DNA, each person can be identified, so you can use DNA, and probably the most famous use of electrophoresis is to get the strands or the bands that show this person versus another, but they're using it for plants.
Et nous avons des exemples ici, après vous allez les voir. On obtient finalement ce qu'on appelle des empreintes digitales, des fingerprinting.
So you can get digital imprints, in this case the DNA fingerprints of the plants as opposed to humans.
Alors nous utilisons évidemment, pourquoi on les utilise dans plusieurs disons, finalités. La première, c'est reconnaître la variété. Reconnaître la variété, cultivar identification, using this fingerprinting as a molecular level, because we have many varieties in the expert, many cultivars.
So do you know how much is a microliter? She's talking about microliters, that's a millionth of a litre, so she's talking about 500 microliters, so about half of 1,000th of a litre.
What's the impact of this mutation? Is it positive or negative on the plant? And otherwise, to study this mutation on electrophoresis genes after PCR. What is PCR? PCR is a polymerase chain reaction. It's a technique that allows to multiplicate DNA, because DNA is very little in plants. You cannot see it. You have to multiplicate it for
Many times.
Yes. You have to multiplicate for study it, and after a multiplication, you can see it in this. In the distance, it's maybe 80 or 70% lectures, and
20% in the laboratory.
Master degree is about the same rate, 50-50.
About 50-50.
Yeah. And PhD is more laboratory.
Okay, and then the doctorate is almost all laboratories.